Good morning, everyone. Thank you all for being here. I am Kellan Weston. I guess a little bit of my background. I just got my PhD from Washington University in St. Louis, and now I'm a postdoc at COMBINEDBrain. And do I point here? I can just reach forward and click it if I need to. Nice, good.
Oh, there you go. All right.
Wonderful. Okay. So what is COMBINEDBrain? COMBINEDBrain stands for the Consortium for Outcome Measures and Biomarkers for Neurodevelopmental Disorders, which is quite a mouthful. But we are a nonprofit that was kind of born out of Dr. Terry Jo Bichelle, who has professorship at Vanderbilt, so we're based in Tennessee. And at COMBINEDBrain, the DYRK1A community has brought us on board to work with them to reach these different objectives that Oliver was just talking about. So we are a nonprofit consortium, and we are led by over 100 different patient advocacy foundations. So we really are a consortium, and we are led by you, the patient advocacy groups, trying to meet what needs each patient advocacy group has. And we basically work to bring together the patient advocacy groups, the scientific researchers, the clinicians, and funding sources to develop a pathway towards ultimately treatments for these rare diseases. So going for the Medical and Scientific Advisory Board objectives, for my talk today, I'm focusing primarily on these three, which I've spent the last few months really focusing on and trying to identify where the DYRK1A community is doing really well, and then where are the sort of gaps and what we still need to focus on. So if you think about drug development and sort of the pathway towards getting a therapeutic for a rare disease, you can start with identifying the gene, right, which we have done. We know about the DYRK1A gene. And sort of if you go along this pathway towards getting FDA approval, towards some sort of drug or gene therapy, there's a lot of different steps along the way in that pathway, where we need to collect natural history data, we need to have scientific models, ID the mechanisms of the gene, we need to interview patients to see what their priorities are, and go towards clinical trials. And if you look at this graph here, everything above the line is kind of what we call, is part of what the patient advocacy group can help with, and everything below the line is mostly what scientists and clinicians are focusing on. So today I'm going to focus mostly on all of these steps that are above the line here. And so COMBINEDBrain can help with all of the different steps of this process, going from diagnosis to helping coordinate disease models and mechanism, coming up with ideas for treatment strategies, working on natural history data, incorporating the patient's voice in our research, assessing different biomarkers and outcome measures, or what are the symptoms that we want to target for this rare disease, and working towards clinical trials. So I've been sort of asking, where is the DYRK1A community doing well? And then where are the gaps where the DYRK1A community can focus their efforts? So just an overview of DYRK1A syndrome. So DYRK1A, you guys are in this really unique position, actually, where the DYRK1A gene is kind of this quote-unquote Goldilocks gene, where if you have too much DYRK1A, this is highly associated with Down syndrome. So, if you have 3 copies of DYRK1A on the 21st chromosome, this is well known to be associated with Down syndrome. But then if you have too little DYRK1A, let's say you have a deletion of 1, or you have a mutation, this is what causes what we know as DYRK1A syndrome. And so this is actually a really exciting position to be in, because the DYRK1A gene has been so extensively studied. So there's actually just a wealth of resources, scientific resources, that are just waiting to be tapped into for studying DYRK1A. A bit of the history of DYRK1A. So DYRK1A, the gene, was first identified in 1995 and in humans in 1996. So really relatively recently. And then over time, we've had more and more patients being identified with DYRK1A syndrome really being identified in its own distinct disease. in 2008, 2011. The Facebook group was founded in 2014. And the very first meeting in 2016, the International Association has been growing since then. And then now in 2024, DYRK1A community, it's been exactly 10 years since the very first DYRK1A Facebook community was formed. And this is really all through the power of you guys coming together and just forming this community and getting your voices out there, starting to get research funded. Over time, the number of known families with DYRK1A syndrome has been increasing. Thank you to Amy for these numbers. And now we have known DYRK1A individuals all over the globe, really almost every corner of the globe, with 801 families being registered just on DYRK1A.org alone, and 315 families in the US, and sort of all over the US, from Hawaii to Puerto Rico, up into Canada. The clinical presentations of DYRK1A syndrome are primarily intellectual disability. We have impaired speech development. Microcephaly are very common problems. Yesterday, we talked a lot about gastrointestinal issues. But as we gain more and more knowledge of who the individuals are who have DYRK1A, the more we can sort of build our range of what are these symptoms that the patients are experiencing. Currently, for management methods, there really are no DYRK1A specific treatments. We have diagnosis and we can manage it. So a lot of individuals will need speech, occupational, and physical therapies. Anti-seizure medications are common. Behavioral therapy, melatonin if there are sleep issues. A lot of individuals use a G-tube for gastrointestinal issues. And also, as we gain more and more knowledge of DYRK1A syndrome, we know that we'll need to monitor for dental, cardiac, and ophthalmologic issues. And also, just want to emphasize the importance of genetic counseling, how often your doctor sort of hands you a diagnosis, but it's really, it's a slip of paper, you don't really know what that means, and how genetic counseling really needs to be pushed forward as a means for connecting clinicians and that diagnosis with the parents and what that means for you as a family. Okay, so DYRK1A Centers of Excellence, Oliver just touched on this, but one thing that the community really needs to establish is Centers of Excellence. So a Center of Excellence is a specialized program, usually at a hospital, within a healthcare institution, which supplies high concentrations of expertise and resources on particular medical areas delivered in a comprehensive and interdisciplinary fashion. So this is really a place where they have expertise on DYRK1A syndrome over and over from DYRK1A community, as well as from other different different rare disease communities. I hear that people have a neurologist, but their neurologist doesn't particularly know about their gene. They're just a neurologist in general, and they want to find where the experts are. So we have one established DYRK1A Center of Excellence, and this is led by Dr. Breji van Von. This is at the Radboud University Medical Center in the Netherlands. So not in the US, we have none in the US. But she has over 20 years of experience, and she's actually one of the very first people to have identified patients with DYRK1A syndrome. So she's really been in this for a long time. So in the US, at COMBINEDBrain, we're working together with the DYRK1A community to really recommend different locations for centers of excellence in the US. So we've come up with some recommendations and different doctors who are there. And these may not be doctors who are experts in DYRK1A specifically, but these are doctors who have knowledge about rare genetic diseases, knowledge about diseases that have a lot of overlapping phenotypes and symptoms, and may be very willing to establish centers where they are willing to learn about DYRK1A and start seeing more of these patients. And here's where I'm going to sort of put out a call to action. So please let us know who your favorite clinicians are. Let us know where your child is going, who their doctors are, even their physical therapists, occupational therapists, speech therapists. Please, you guys as the community know best who's good, and please let us know so we can start contacting these people and see if they'd be willing to be more involved in the larger DYRK1A community. The second thing that needs to be established are standards of care. So standards of care are protocols for best practices for monitoring DYRK1A syndrome patients and prescribing therapies for DYRK1A. And these are standardized across into clinical centers worldwide. So especially because DYRK1A is still being understood as a gene and as a disease, there really is no standard of care that's established for DYRK1A syndrome. There's no easy protocol that your neurologist or your doctor or your pediatrician can go to and say, okay, step one, look at this. Step two, assess for this, and maybe consider these different drug treatments or these different therapies. And so just here is an example of a standard of care, a graphic version of a standard for care. Specifically, this was for seizures in Dravet syndrome. This is the gene SCN1A. And sort of just an easy, almost protocol for clinicians to follow in terms of what to monitor for in these kids, what to look for, what to prescribe if the kid has this symptom, or what to prescribe if the kid has that symptom. And so as we establish, I'm going to go back, as we establish these centers of excellence, this can go hand in hand with establishing standards of care. So at COMBINEDBrain, our plan is to work with the DYRK1A community to identify the clinicians who have expertise in treating people, and also the clinicians who have just expertise in treating people maybe with other rare genetic diseases with overlapping symptoms. We're going to coordinate discussions to establish these standards of care, and then distribute the standards of care to the different centers of excellence and just across the states and across the world. Here's just a slide on family support resources for DYRK1A families. Many of you may know these. These are all links, so if these slides get shared, you can just go ahead and click on them. But I just want to highlight the Simon Searchlight community. I know that Helen Wilsey, who spoke yesterday, one of the motivations for, or let's say inspirations for her research, was seeing that just how many of these patients have gastrointestinal issues, and she was getting this via the Simon Searchlight community. There's the DYRK1A Facebook group. I just want to highlight the Connected Parent, which was just founded by a different patient advocacy group leader for the PCDH19 Foundation, his resources for parents. Yeah, all right. So now I want to talk a little bit more about what COMBINEDBrain has been doing since our meeting last year. So we've been working on this disease concept model of DYRK1A syndrome, and this is being led by our fearless Olivia Solverson here at COMBINEDBrain. And she is a master's student in genetic counseling, and she's doing this project as her master's thesis project for genetic counseling. And she's asking, what are the symptoms that are most frequently reported in the published literature? And so she's done a deep dive into PubMed, which is where you find most of the scientific literature, and has categorized which symptoms are the most reported, sort of which symptoms are clinicians seeing and reporting the most. And I think for a lot of you parents, these may come as no surprise, but we have intellectual disability and developmental delay as some of the most commonly reported symptoms. Seizures are very high up there, as well as, oh, I don't see it anymore, gastrointestinal issues, autism, even cardiac defects or GI problems, scoliosis have even been reported sometimes as well. So that's the literature and the published literature and what doctors have seen in the clinic and actually taken their time to report on. But we want to make this even more patient-focused. This is getting information from doctors. Now we want to get information from you guys, the families. So over time, the FDA has established a sort of codified a way to identify what is important to patients, and that way to look at what clinical outcomes that the patients want to see as a result of a given therapy. And over time, this has been honed, and now we're really trying to incorporate the patient's voice in medical product development and regulatory decision-making. And here's an example of one for Angelman syndrome, another rare genetic disease that was published fairly recently. This is a little bit small, but this is sort of the output of what one of these disease concept maps do. And we can look at communication, neurological behavior, cognition, and motor difficulties. It's asking questions about how well do the kids sleep? What are their gastrointestinal issues? Even things like, do they have skin problems, visual problems, emotional problems? We also look at impacts on the patient, so community and school, socialization and family life. We also look at caregiver and family burden. So, I mean, does this affect the family's sleep schedule? Does this affect the family's financial burden, the parent's anxiety? Things like that. It's really a comprehensive view. And so this is done via interview data, where we ask what are the lived experiences of the person and their family, what matters most to them, and what symptoms should we focus on for therapy development. And so Olivia is starting doing one of these disease concept models where she's using her findings from her literature review to inform in-depth interviews with the parents. These are interviews that last about 1 to 1.5 hours. And with this information, I think I texted her last night, and she's done 5 families so far, but is still recruiting. Hopefully this will turn into a holistic sort of graphic image of what matters to families. Anywhere from symptoms, behavior, cognition, symptom impacts, so the autonomy of the person with DYRK1A, schooling, sleep, socialization, and also caregiver impacts, so daily life activities, emotional impacts, family life, leisure, support, what matters to the patient and to the caregiver. And Olivia wanted me to highlight that we are still recruiting, so if anybody would like to participate in this, she's still looking for DYRK1A individuals to really get this holistic view of what DYRK1A syndrome is and what it's like for people. This will ultimately go into a publication. So this top graph here is actually from a very recent publication that we at COMBINEDBrain helped to put together. All right, the next issue is understanding the genotype-phenotype correlation in DYRK1A syndrome. So when you go into the clinic, let's say you, your kid just got diagnosed with DYRK1A syndrome. So the doctor hands you the genetic report. It's a lot of letters and numbers. You're not really sure what it means. And honestly, often even the clinician is going to have trouble predicting how severe that outcome is going to be for that individual. We can predict, but it's difficult. And so to predict pathogenicity, which is just a way of saying, will this genetic mutation cause disease? And if so, how severe will that be? We often turn to ClinVar, which is a publicly available database where doctors actually log all their different genetic diagnoses of their patients. However, even that is not the best predictor, especially for these rare genetic diseases. So if we just look at DYRK1A mutations in ClinVar, about 41% are classified as benign or likely benign. So individuals may have variants in DYRK1A that cause almost no symptoms. 30% approximately are classified as pathogenic, just meaning it will cause symptoms. But I really want to highlight here where 26%, so a good quarter of the variants that are logged in ClinVar have uncertain significance. We really don't have a way to predict what they're going to cause for that person with the mutation. And then second, for predicting the severity of DYRK1A syndrome symptoms based on your genetic report, there really is no great way to do that. So how do we gather data towards trying to understand this for DYRK1A syndrome? So if you look at the DYRK1A protein, this is the protein sort of laid out. If you think of a protein as sort of beads on a string, you've laid out the string in one line. So if you look at the different amino acids, which are the building blocks of a protein along the x-axis here, then the y-axis here is the number of genetic mutations logged into ClinVar. There's really no hot spot, if you will, of genetic mutation. So there's really no easy way to say, okay, if you have a variant here in this catalytic domain, it's going to cause more severe phenotypes. Or if you have a variant here in this nuclear localization sequence, you'll probably be, your symptoms won't be as severe. So our aim is really to identify the different protein regions and amino acids that are less tolerant to mutation. So does a deletion versus just a missense mutation cause different disease outcomes? And do mutations in certain regions of the gene correlate with disease outcomes? So if you think about the mechanisms of how you can lose function, so if you have the full-length protein, this is going to be folded up into a functional DYRK1A protein. You can have a whole gene deletion where you just have no protein, so you have less DYRK1A. You can have a missense mutation. This is where just one of those building blocks is wrong. So in this case, example, this is a lysine to an isoleucine. So one building block is just the wrong, it's been swapped for the wrong building block. And so then that gives you a mutated protein. And a mutated protein can be benign, it can be loss of function, or it could even be a dominant negative gain of function. This could do anything, and it's really difficult to predict what just one change will do. You could also have other mutations, such as a nonsense mutation or a frame shift mutation, which can lead to a truncated DYRK1A. This is usually loss of function, which often gets unstable and gets degraded. And it's important to understand the differences between these different mutations for scientists, because these will likely need different therapeutic strategies. Another way to think about this is that you can have a loss of protein level, so you don't have enough DYRK1A. You can have a change in its activity, so DYRK1A is unable to do its job in the cell, or you can have it be mislocalized. It's not in the right spot, so it's probably not going to be doing its job properly in the cell. And again, this is important because these different mechanisms, which somebody may have one or multiple even, will cause different symptoms and will probably need different therapeutic approaches as well. So towards that combined brain, we've been looking at the published DYRK1A syndrome cases and looking at sort of, at least in the published literature, so these are when clinicians actually see patients with DYRK1A syndrome and publish on that. So these are probably likely to be patients who have more of those symptoms, looking at what kinds of mutations they have. And a lot of them do have these frame shifts or these nonsense mutations, which really shows that DYRK1A, the mechanism often probably is a loss of DYRK1A. And then sort of upcoming, we are at Combined Brain, we're collaborating with Dr. Dennis Lal. He's at UT Health, and he's working with the Broad Institute to make a portal. It's basically a one-stop portal for a lot of these rare genetic diseases, where it aligns all the different reported mutations along the length of the gene. And you can actually look in three-dimensional space at where those mutations are on the gene. And this would be a great resource for scientists to take a look at where are the mutations, what kind of mutations are happening, and also this portal will have patient-reported phenotypes as well. So what symptoms are the different people with different mutations experiencing? All right, so now I want to go into drug development resources. So our first drug development resource, or sort of think of it as again, that pathway towards drug development, are our leaders in the field, our clinicians. So here we have some clinicians with expertise in DYRK1A. We also have our medical and scientific advisory board at the DYRK1A community, as well as many different researchers across the globe who are working on DYRK1A. And DYRK1A is really in a unique position because I'm actually really impressed with how international this community is. Most of our rare disease groups, they're really just very isolated. So there's a US group, and then maybe there's a Spain group, you know, but they're not talking to each other. So I'm really impressed with how well just truly how international and collaborative the research is on DYRK1A. We have, cell and animal models, I'll just touch on this, but DYRK1A is also in a really great position in the fact that we have really good cell and animal models. So a model can be used for answering the basic questions about how DYRK1A functions, and it can be used for preclinical experiments and testing therapies. You can have in vitro models, these are cells, and in vivo models, these are animals. And I just want to highlight specifically cell models right now, because our cell models, especially for DYRK1A, are really fueled by you, the patients. So this is a slide from Simon's Searchlight, where blood cells obtained from the patients can get turned into specialized cells, induced pluripotent stem cells, and then differentiated in a dish into different cell types to study what DYRK1A mutations, what effect they have on different cell types in the whole body. And they can also be used for drug screening and discovery. And this research really is fueled by each individual's variant. And I just want to highlight, this is Simon's Searchlight. These are all the different iPSCs that are available through Simon's right now to researchers, whoever wants to request them. And these are all variants that were provided by you guys from blood samples, from tissue and blood collections. Animal models, all I'm going to say is that we are in a great position with animal models because DYRK1A is highly conserved, which means that the gene in rats, mice, frogs, flies is very, very similar to the human gene, which makes them a really good model for studying what the effect of different mutations are going to do. And also, again, because DYRK1A has been so well studied in Down syndrome, a lot of these models, there are a lot of labs around the world who've studied DYRK1A in Down syndrome and could be even used to study DYRK1A in DYRK1A syndrome. Next, I'll talk about natural history. So DYRK1A patient registries, sort of looking at where are all of the patients right now. Simon's Searchlight is a very good tool for that. There's 156 patients enrolled as of this April, and they have their biorepositories. That's those blood samples that can be differentiated into cells available for scientists. COMBINEDBrain matrix, I think last year at this group, members of COMBINEDBrain came and took, I think, blood and tissue samples. And from that, we have 19 patients with I don't think that adds up. Anyways, 19 patients with, we have 20 participants. I know this number is correct. 20 participants with 7 controls. And I'll talk a bit about where the data generated from that is going, which is very exciting. And then you in the DYRK1A community really use RareX as your primary registry. I know, I don't have the current numbers, but I know there are at least over 50 patients registered on RareX. And this is really where we can go to say, okay, who are the members in this community and what are their symptoms? What are their genetic reports? I just want to highlight right now, sorry, I haven't been holding this up high enough, highlight right now that if you are in RareX, to please look and see if you have a CRID, which is a clinical research ID. And the CRID is how we can pull your de-identified genetic information from RareX into other portals, such as the COMBINEDBrain portal, for research purposes. I believe Simon's also might use the CRID. I have to fact check that one. All right, so those are patient registries. So now let's talk about natural history. There's a lot of vocabulary today, sorry guys. All right, so a registry is a point in time. So say your child gets diagnosed with DYRK1A syndrome at the age of 3. And in a registry, you can go into, let's say, RareX, and input your child's name, your contact info, your diagnostic information, your medical information. And that's really great. We know who's out there, who has DYRK1A syndrome. Natural history, however, is longitudinal, which looks at the course of DYRK1A syndrome over the course of these people's lifetimes. And natural history can ask questions such as, will the child have seizures in the future? Will a certain medication help more than others? When will gross motor skills develop? And when will the child learn to talk, learn to walk, all of these things? And that way we can sort of look at the development of DYRK1A syndrome over time, which is something which we don't have a lot of information on right now, especially as a lot of these people age. And so natural history studies, these can be conducted in a very systematic way. So Simon's Searchlight is doing their natural history study. This is an example of a lot of the Simon's Searchlight data that's been generated. There's the Tiger study. Emily Newhouse, I think, is speaking after, hello, you're speaking after me. I won't, yeah, I'll let you talk all about this. And, but yeah, this is another natural history study going on right now. And then RareX, as we have more and more patients complete surveys in RareX, especially if any of these are completed. I'm not sure exactly how the DYRK1A RareX is set up. If it's set up to complete surveys over time repeatedly, this can really show the trajectory of DYRK1A syndrome over time. Okay, finally, I'll be talking about omics data of DYRK1A syndrome. So you may or may not have heard of any sort of big word that ends in the word omics. So we've got genomics, epigenomics, transcriptomics, proteomics, metabolomics. These are all ways of understanding what different genetic gene mutations are doing, sort of on a broad scale level of all the cells in your body, how it affects your DNA, how it affects the methylation, that's the regulation of your DNA, how it affects the RNA, what Craig called the software yesterday, metabolomics, what sort of different small molecules in your body are changed with DYRK1A syndrome. And then today I'm going to talk especially about proteomics, because at COMBINEDBrain right now, we're working on a really exciting project with proteomics, where we're asking what are the different protein expression changes in DYRK1A syndrome. So we've collected blood samples from 27 individuals. We have 19 DYRK1A individuals and 8 healthy controls. And we're working together with SomaLogic to conduct proteomics. And proteomics is really cool because you can take a look in this graph here. In blue are proteins that are downregulated. So in this case would be downregulated specifically in DYRK1A syndrome, or upregulated. So which proteins do people with DYRK1A syndrome have too much of or too little of? And can we sort of establish a certain protein or set of proteins as a biomarker for saying, here's how we can recognize people who have this certain type of disease? I just want to highlight right now that I think these were all, before I joined COMBINEDBrain, collected before I joined COMBINEDBrain last year at the conference. And I just want to highlight that 10 individuals are missing their genetic reports. So if you have donated any tissue samples, please reach out to me or Anna Falzer at COMBINEDBrain. We would love your genetic reports, because if we don't have your genetic report, we can't analyze your data, and we'd love to get as much data as possible. And also, we want to be able to compare your genetics with your proteomics and also your symptoms. So we want to have your genetic report, we have your proteomics, and then we want to know the symptoms that your child has. And so I know on RareX, often you're filling out a lot of surveys. The Vineland or the PELS are 2 of the common ones. But again, we need your CRID, your research ID, in order for us to pull this information from RareX. So if you have any questions about the CRID or how to create a CRID, it takes all of 60 seconds. I've created one for my younger sister who has a rare genetic disease. I can help you through it. All right, and then finally, just conclusions and recommendations for families. So going back to our objectives, COMBINEDBrain, again, is helping to deepen the understanding of the individuals affected by DYRK1A syndrome. We're working towards establishing a drug development toolkit, working towards a path towards therapeutics, a path towards clinical trial readiness. Do we have all of the patient information, the scientific information that we need to start a clinical trials? And then defining centers of excellence. Who are your doctors? I think I just said all of that in the last slide, but working towards finally clinical trials. So in conclusion, DYRK1A syndrome is increasing the number of patients that we've identified, which is really exciting as we identify more and more of these people all around the globe. There remains a lot of opportunity to expand on the existing knowledge. We can fund further critical research, and we can support the scientific and medical community. There's a lot of fundraising opportunities for us to support these scientists. And then COMBINEDBrain, this is my job as the Rent in Neuroscience. I've done a landscape analysis to sort of look towards recommendations for what can be pursued by the DYRK1A Medical and Scientific Advisory Board. And then finally, just recommendations, because I'm at a family conference. So here are my recommendations for you, the families. So please tell us who your favorite clinicians are so we can work towards establishing centers of excellence. In RareX, please make sure you have a clinical research ID in there. And let's try and get as many genetic reports as we can, as well as filling out the different surveys, such as the PELS or the Vineland. The disease concept model, these are those in-depth interviews trying to really see what the impacts of DYRK1A syndrome are on the families. Please contact Olivia or me to interview. Natural history studies, which we'll hear more about very shortly. These are also opportunities for enrollment. And social media. I've seen that on social media, the DYRK1A community is really, you guys are doing great. So keep it up. Keep advertising DYRK1A. You're doing great things. So thank you to the DYRK1A syndrome community as well as to COMBINEDBrain. And thank you, everybody, the families and patients.